So welcome to video 6 of this course G05, where we're going to look at epitope-specific antibody profiling using the theory behind that before we look at the demonstration video later. So this whole approach is based on what's called BBEA, which is B-based epitope assays that allows us to look at the peptide specificity of different antibiotic profiles. And here we're looking at that in the context of IgG measured in human blood sera. And you can look at this website for more background that contains more examples of what we're doing here. So we're kind of looking at an instance where we have cross-linked IgE that is bound to an allergen. In this instance, binding to a particular egg protein, and that IgE activates mast cells and basophils, and this creates the allergic reaction of having high levels of histamine, heparin, and tryptase release. And our goal then is to look at the specificity of IgE to different epitopes of this egg protein to understand better the clinical reactivity and allergy severity. So what we're looking at is this bead-based epitope assay that creates numerous epitope-specific antibodies and their segments of their peptides. So in some ways, the analysis steps are like gene expression analysis, where we import our data, we do QC and counting, we look at we normalize it, and then look at the effects, and then try to interpret the patterns at the end in terms of differential antibody binding. So what we're looking at is peptides from the ovomucoid protein, which is associated with egg allergies in humans. And our key question of interest is which parts of this protein does IgE bind onto? So what we're doing to tackle this is to split the IgE amino acid sequence into 58 parts, each with 15 amino acids, the 12 amino acid overlap between different parts. And that's tested in 38 children, including and also 6 controls, giving 44 altogether. And we're looking at data from a Luminex 200 assay across a 96 well plate, where we add to those 44 by doing them in duplicate, and then we have 2 positive controls and 1 negative control as well, and then also 2 empty wells against which we can correct the background rates. So the steps here are that we're going to do library installation and setup, do QC normalization, QC of the normalized data, looking at the similarities across the replicates, the average across the technical replicates, and then do differential analysis of the relative binding to the different peptides of interest. It's worth noting the normalization approach here. So for each epitope-specific antibody J in sample I, MFI is normalized as this formula, where yi is the log2 scaled value plus the offset. So the offset should be some small value like 1 to prevent taking log2 of 0. That's our log2 scaled value, yi, and yi goes in here. And we're saying that if yi is above the background rate, so this is the difference between yi and the background rates, and BG is the background empty cells. And there we're summing across the values of yi for the samples to get that normalized value for the yij of interest. And we were able to get this NMFI value at the end, where that's set to 0 if the background intensity is higher. And that allows us then to take this normalized data and use a limit to analyze that further. So that allows us to explore these epitope-specific IgE antibody peptides using commands that you may have experienced before looking at gene expression analysis. So that's where we're looking really at each linear model containing each feature's rate y and modeling it in relation to the phenotype x of interest. So in Notion, has some sort of y is equal to b by x plus some sort of error. Notably here, we're pooling the variance across different features with similar rates to account for the fact that the sample size for a particular peptide of interest may be small, and that we're also using a negative binomial distribution, which is a generalization of Poisson and geometric distributions. So we're assuming here for limit that our residuals are normally distributed, that we have a single outcome, that the analytes are independent here. That would mean the same if we're looking at genetic or transcriptomic data, that the genes are independent, that the samples have equal weights, and that we are dealing with a negative binomial distribution as well. So we can ask, is the data log normal and has to be scaled by a factor as well to prevent taking the log of a 0, for example. So let's look at this in more detail. So we have this overview here where we have a matrix of expression values as shown here, and then we're using these linear models to infer the expected value for yj and use that to infer x by beta j and get the variance of this across the different samples to scale that by weight accordingly. So for each gene G, we have an expression values and a design matrix X associated with that, associating those values with the coefficient expression beta G for that gene. We're using empirical based methods to get a better posterior variance estimates. That's SG squared star over here on the right. It adds observation rates for each gene G for each sample J to adjust for differing data quality, and then it uses variance modeling to accommodate technical biological heterogeneity as well. So there's quite a few steps here. That means that our initial input data and our output data might differ by somewhat. So in terms of the steps of how we go about this and implementing these models, is that we have to firstly make a design matrix and then define the comparison we want from that design matrix. We might have multiple different groups we want to compare. We need to define what contrast for each comparison that we want to make. We fit a linear model based on this comparison and design matrix. We fit a contrast model from that. We're able to use empirical ways to moderation to get the features standard errors, and then extract the differential analysis metrics, just the log two-fold change, adjust the p-value, and so on at the end. In terms of this step of the design matrix and the contrast, it's worth noting that there's two kind of models. The more simplistic one, so if you have a gene J where the x is a binary value of 0 or 1, we can simplify this as simply yj g is equal to b0 plus b beta 1 by x. So that means either x exists or it doesn't, depending on the category. So in this case, beta 1 denotes the changes of interest. For a more advanced one, we have two different groups where you have groups A and B associated with gene G with x0 equals 0 for group A and x0 equals 1 for group A and x0 equals 0 for group B. And in contrast, we have x1 which is 0 for A and x1 which is 1 for B. And thus when you have our formula here of yg is equal to beta 0 x0 plus beta 1 x1, we can have our contrast between beta 1 and beta 0 as denoting the changes for each of these two groups A and B. So please see the demonstration video on epitope-specific antibody profiling with R for more details.